Okay, recent breakthrough. Just coming out of the lab last week, the direct vascularization of a tumor. So we always had problems that the tumors were destroying the matrix around them, so we couldn't get a vasculature growing into the tumor, and now it looks like we are able to stabilize that and really create a vasculature which directly grows into little tumor spheroids. Hey, my name is Tom Sommermann. I'm one of the senior scientists here at Dynamic42, and I'm heading the cancer research team. I was attracted to science probably because of my high school teacher, big shout out here, and he showed me how amazing biology can be, looking at cells, looking at genes, looking at the behavior of organisms, and that attracted me to science, starting to study biology. And then I got very early into cancer, looking at colon cancer initially, and then later mostly lymphomas and the immune system behind that. And that excited me, and this was something I could continue here at Dynamic42, and so I'm very excited to now work on that problem for more than 10 years. My professional experience is mostly in cancer immunology and generally the interplay between tumors and the immune system. I started very early at the University of Erlangen-Nuremberg looking at colon cancer, then transitioned to Harvard University, where I was looking at B-cell lymphomagenesis and how viruses play a role in how we get B-cell tumors. And then I did 9 years at Max Delbrück Center in Berlin, where I was looking at transgenic mouse models of B-cell lymphomagenesis and how the immune system reacts to these B-cell tumors. And then I eventually transitioned to Dynamic42. Coming from the field of mouse modeling and working with transgenic mice, I was always interested in these models which could bridge the gap between general 2D culture, simple experiments we were doing in the tissue culture, and then the very complex mouse models we were running, which were highly expensive and of course took a very long time to license. So I always thought these 3D models would really fit there to bridge that gap. And working at Dynamic42, I'm now very excited to actually work and develop to help bridging this gap and find new ways to really make complex modeling available for tissue culture and not only for mouse models. Oh, most exciting projects. I mean, there's a plethora of projects we are working on here at the moment on the cancer team. Certainly, we just published a pancreatic duct adenocarcinoma paper, which showed for the first time that we can use these microphysiological systems, our organ on chip, to really show also immune invasion into these tumors, see that we can treat these PDAC tumors with drugs and see how they are going over a vascular barrier into the tumor. So this is very exciting, but also some other work where I'm allowed to look at neoangiogenesis and kind of build vascular networks in a chip. And this is all very exciting work and amazing what one can recreate in such a small organ on chip. Okay, recent breakthrough. Just coming out of the lab last week, the direct vascularization of a tumor. So we always had problems that the tumors were destroying the matrix around them, so we couldn't get a vasculature growing into the tumor, and now it looks like we are able to stabilize that and really create a vasculature which directly grows into little tumor spheroids. That just looks amazing because you can see this 3D network interacting with this tumor sphere, so this is very exciting. I'm absolutely certain within the next, let's say, 10 years, these 3D models, organ on chip or other microphysiological systems will become more and more important in the drug development process. Just think about that we are developing more and more targeted drugs which really look for the one specific molecule, especially coming from the immune system, for example, antibodies, really targeting that one molecule in the human that might target your cancer cell, that might target your disease cell. And we have to see that these targets will not always be or rarely be conserved in animals. And so we will have to find new ways to screen in a human system for function of these drugs and function of these cellular therapies. And I think organ on chip or other microphysiological systems will be the answer to that problem. A common misconception about organ on chip. Well, I I think a common misconception is that people think it's just a new, novel way of kind of fancy culturing your cells and make maybe a few pretty images to convince a reviewer that you did a cool experiment. But what I really want to stress is organ-chip technology is really changing the biology of the cells you're putting in. They behave very differently in a 2D culture versus in a 3D culture with perfusions, shear stress, stretch and strain stress. So cells start having different expression profiles. They start behaving way more like they behave really in the body. So organ-chip really matters if you want to create a model system that more resembles the in vivo setting than normal 2D.